ricin, which is a protein that comes from the castor oil plant. A small dose of ricin can be fatal to humans. The information found on our page is important because as of now there is no cure for ricin toxicity. Hopefully with further research of the mechanism that ricin uses to attack cells, a cure for ricin toxicity can be found. Here in our compound info you can see adenine, which is depurinated during the mechanism, and arginine, which is the general acid catalyst for the mechanism. Ricin has two subunits, the A chain and the B chain. The A chain contains the active site and the B chain contains the binding site. The A chain is responsible for the depurination of adenine, leading to the deactivation of the ribosome. The B chain has a binding site that attaches the ricin to the cell. After dissociation of the two chains, the A chain enters the cell and performs its attack. Once ricin attacks, protein synthesis in the cell stops and the cell dies. Next, we'll look at the challenges for this mechanism.
According to empirical evidence gathered in the papers we looked at, the mechanism of action of ricin is SN1 based. This mechanism, so the SN1 based mechanism, as first the adenine base is catalyzed as a leaving group by the arginine residue in the active site that acts as a very good general acid catalyst, and it forms actually carbenium ion intermediate, which subsequently gets attacked by a molecule of water, and then this arginine residue becomes regenerated by deprotonating the water attached to the carbonyl carbon of the former oxycarbenium ion intermediate. And also, just so you can get a look at it, you can get a look at the SN2 mechanism that was thought to happen for a while until experiments proved it was oxycarbenium ion intermediate, and well, disprove this. And obviously, the one thing I want to take away from this mechanism is that there's no oxycarbenium ion intermediate anywhere in this picture. And also, for a while, it was thought that ricin acts as a phosphorylysis enzyme, which means it uses a phosphate group in the place of water to essentially do the SN1 type mechanism. And that's the really only big difference between this this step and the one that happens in the water is that the phosphate replaces the molecule of water.
Why does the mechanism proceed with arginine as a general acid catalyst? Under physiological conditions, arginine is positively charged, lending as a good electrophile for attack of the N3 nitrogen of adenine. We analyze the hypothetical substitution of tyrosine 80 and glutamine 177 for arginine in the mechanism. Here's a JMOL model of the active site of ricin that includes arginine catalyst in light green, along with tyrosine in red and glutamine in light blue. Glutamine does not have a hydrogen available for attack with N3 and adenine, and tyrosine would result in a less stable tyrosine intermediate. Also, here are marking sketches showing only the first step of the reaction where the respective amino acids are used, hypothetically, or the catalyst in the ricin mechanism to depurinate the adenine. Here is arginine, and then here is tyrosine.
To prove that arginine is critical to the function of the enzyme, mutants of arginine were produced to see how the results would differ from the wild type. When arginine 180 was replaced with histidine, the kcat decreased by 1,000-fold. When arginine 180 is replaced by lysine, kcat decreased by 4 times. Base stacking of the A chain with adjacent adenine, guanine, tyrosine also help stabilize reactants and intermediates. Based on X-ray crystallography, tyrosine 80 and tyrosine 123 in this JMOL image here interact with adenine through pi stacking interactions. Glutamine 177 also stabilizes the oxocarbenium intermediate of the ribosyl ring. These pi orbital stabilization interactions are important because stabilization of key intermediates are what drive the reaction forward. This makes the depurination reaction more likely to occur. Thank you for watching our Molodeck, and remember, don't take ricin.